All right. So isoelectric focusing is separating proteins by charge, so specifically by its isometric point or pi. So, what is pi? The isometric point is the pH at which a protein has no net charge. So, how pi or IEF works is proteins or isoelectric focusing, proteins are loaded into a polyacrylamide PIE or IPG strips and are fixed with a fixed pH gradient. So it goes from one pH to another. And an electrical current charge causes the proteins to migrate to their pi. Movement stops when the net charge of the protein is zero. Applications, so we can use high resolution protein separating, and it's the first step in 2D protein electrophoresis. So, a two-dimensional page. So, step one is going to be the IEF that we just talked about. And this is where proteins are separated by their isoelectric point using that IPG strip. Then we run an SDS page. So, the IPG strip is placed on top of an SDS page gel. The proteins migrate to the gel and then are separated by size. So, the outcome of this is each protein appears as a spot on the gel, and then it's one gel per sample. The spot patterns are analyzed with a software for comparison. And then we can identify these spots by analyzing those spots with mass spectrometry. So, finally, we can look, or we've got a couple of others. So, next we can look at nucleic acid separation. So, we can take high resolution DNA fragment analysis and separate the protein out by their DNA fragments. So, ideal for protein fragments less than 1 kilobase. And we use a TBE buffer or a trisporadic EDTA buffer. And it uses sequencing gels, so specialized polyacrylamide gels that resolve DNA into single base pair differences. And then we use a manual DNA sequencing to get these separated. So the modern technique is called capillary electrophoresis, and it uses polyacrylamide in thin tubes, and the DNA fragments are separated by size. We can detect automatically as they pass through a laser or a detector, and it was the foundation for automated DNA sequencing. So now we can use capillary electrophoresis to automate DNA sequencing. And then we have protein chromatography. So it separates biological molecules from complex mixtures. So, what's the purpose of this? And it's used for purification of proteins, especially recombinant proteins from bacteria cells. It removes bacterial proteins, lipids, and carbohydrates from whatever sample we're working with. So, the base components of chromatography is the modal phase, where in a liquid buffer solution, and then a stationary phase within a solid resin bead packed into a column. So, column chromatography setup, we have resin beads used specific that have specific chemical properties tailored to the type of chromatography being used, and a buffer flow over this resin, allowing separation based on molecule interaction. So, why is this important? What are the applications? So, it's essential in protein purification, biochemistry, and biotechnology workflows. So, how do these molecules interact with stationary and mutable phases? So, molecular affinity, Molecules with stronger affinity to the stationary phase migrate slowly through the column, while molecules with a weak affinity for the stationary phase move faster. So this chromatography process uses simple solutions containing proteins is loaded into this column. As the solution flows through the resin beads, the molecules bind to the resin or pass through. The solution is then collected in functions down at the bottom. And then we have elutions and washing. So the washing step removes proteins weakly bound to the resin, while the elution step or identification step uses a different solution to release the proteins from the interest of the resin, and then they can collect the fractals on the different washings, and they are analyzed to determine which contain the protein of interest. So does the first one, none of the weak ones, or the strongly bound ones? And then we have size exclusive chromatography. So molecules are separated based on size. There are porous beads in a resin that allow small molecules to enter, but not larger ones. So, how this works is large molecules are excluded from the pores and move quickly through the column, while smaller molecules enter the pores and slowly move through the column. We get fractalization, so proteins are separated into fractions according to how fast they flow through the column, which correlates with their size, right? The smaller ones are going to move slower than the faster ones, and we can commonly see this used for separating proteins, polymers, and other large biochemical molecules. So we also have hydrophilic interaction chromatography, and this is where proteins are separated based on their hydrophilicity. So are they water repelling or water philic? So do they like it? Or hydrophobic? Do they not like it? So hydrophobic proteins have more hydrophobic amino acids on their surface, making them repel water. So how this works is high salt conditions cause proteins to bind to the hydrophobic resin in the column. As salt concentrations decrease, proteins are gradually eluted from the column based on their hydrophobicity. So if they like the water, they're going to disappear. And if they don't like the water, they're going to stick onto those beads and not come off. So hydrophobic proteins bind more tightly to the resin in the high salt environment and require a lower salt concentration to elute. Elution order, so most hydrophilic proteins elute last. Hydrophobic, sorry. The ones that don't like water let go fast after the salt concentration is significantly reduced. This is often used to separate proteins, enzymes, and other biological molecules based on their hydrophilic interactions. And then we have ion exchange chromatography, and this is where molecules are separated based on their net charge, so positive or negative. So, types of ion exchange resins, we have cation exchange resins that negatively charged molecules bind to negatively charged resins bind to positively charged molecules, cations. Anion exchange resins are positively charged resins that bind to negatively charged molecules or anions. And samples are loaded into a column at a in low salt buffers, and molecules with opposite charges to the resins bind to the column, while molecules with the same charge flow through it. Binding is reversible, right? Proteins can be removed by gradually increasing the salt concentration. Proteins are released from the resin based on their charge by adding a buffer with increasing salt concentration, disrupting the charge interaction. We can use this for separating proteins, nucleic acids, and other biomolecules with different charge proteins. And then we have affinity chromatography. So, a principle of affinity chromatography are molecules are separated based on their affinity for a specific binding pattern. Resin beads are coated with ligands that have high affinity for a target protein. The protein of interest binds specifically to the ligand, while other proteins in the molecules flow through and are washed away. So, samples are loaded into the columns and proteins bind. The non-protein target proteins are washed away and the targets are eluted by changing the charge conditions. So we can see this used in purification of specific proteins, such as recombinant proteins or proteins with known ligands. And this is commonly used to isolate manufacturer proteins from complex mixtures, which is super helpful for industries that produce insulin or other things where we need to specifically pull that protein. It's highly specific, making it one of the most effective methods for isolating a single protein from a mixture. Ligands used in affinity uh, chromatography are typically antibodies bound. They bind specifically to target proteins, cofactors that assist in binding proteins that require them for activity, enzymes or substrates that bind to a specific protein target based on the enzyme activity. We can also have recombinant protein tags. So the HS tag is an example of this. It's a short sequence of histidines that bind to nickel ions on the resin for an effective protein purification. So, how these ligands work, these ligands are immobilized, so stuck to resin beads in a chromatography column. Proteins of interest bind to these ligands while other proteins are washed through. And there are two elution methods for the affinity chromatography. So, we have a competitive elution, so adding another molecule that binds more strongly to the ligand than the target protein, forcing those proteins of interest to be released. Then we have a pH reduction, so lowering the pH alters the protein's structure, reducing the affinity to the resin, leading to them being washed off. So, applications of this process: histag affinity chromatography of purifying recombinant proteins. We can also see purification of antibodies, enzymes, and other biologically relevant molecules for industry as well as healthcare. So, analyzing the results of chromatography. So, methods of analyzing fractions. So, once we have separated out all of the different proteins, we can run an SDS page. We can run an enzyme assay or a protein concentration determination. So we can use the SDS page to analyze the protein composition of each fraction and visualize protein purity and molecular weight. It also helps confirm the presence of a protein of interest. Our enzyme assays are used when purified enzymes or other functional proteins. The assay measures the activity of a protein in each fraction to confirm that the protein of interest is still functional. And then protein concentration determination. Once again, we're going to use spectrometry, right, measuring the absorption, or protein quantification assays, like the Bradford assay that we talked about up at the beginning, or at lesson 2. So, analyzing chromatography system monitoring, there are specialized systems like the NGC chromatography system, and they can measure conductivity, so tracks the ionic content of eluting systems, useful in ion extraction chromatography or the absorption, and it monitors specific wavelengths to detect the presence of proteins or other molecules. And why these monitoring systems are important: it assesses the purity of proteins, it confirms functional activities of proteins, and it quantifies the protein yields and monitors the elution process. So, how do we scale chromatography, right? If this is important, oh, I forgot to delete this. Ooh, well, let me skip that slide. So, um, right. So, small to large scale processing chromatography, column diameters are typically a meter or larger. When we use them in manufacturing, it is common to in the production of industrial enzymes, biological drugs, and large scale protein purifications. Performed in large scale, highly controlled and automated systems. The principle remains the same as the smaller ones. The chromatographic separation principles are consistent at all scales, either small or high. But the highly automated systems are tightly controlled to ensure efficiency, careful optimization of flow rates, buffer conditions, and elution protocols for larger volumes so nothing gets messed up or screwed up. Key considerations for scaling up: we can see maintaining uniformity and consistency in separation as column size increases, ensuring reproducibility in both batch to batch and scaling up processes. And then we can control system variables such as pressure, flow rates, and loading samples. So make sure everything is controlled, uniform, and reproducible. So protein analysis using bioinformatics. So, bioinformatics and protein sequencing analysis, so we can have the exploration of raw data. And this is advances in biotechnology and computing have produced a massive database of protein sequences. So, what is bioinformatics and what does it do? Using um, computational tools to compare multiple sequences quickly and accurately. Also, it identifies similarities, differences, and evolutionary relationships among proteins. Some key applications are sequence alignments, so determining how closely related one protein is to another. Conserved regions, so identify amino acids or motifs preserved across species, which is essential for protein function. Functional predictions conveys areas, conserved areas may suggest active sites or critical structural elements, and then molecular evolution, so it helps trace evolutionary pathways and the origins of proteins. We can also see so, impact on the bioinformatics: it guides mutation or mutagen studies and informs drug design and protein engineering, and it supports annotations of genomes and prediction of unknown protein functions. Protein sequencing and alignment in phylogenetic analysis, so sequencing alignment tools. And those tools like cholesterol omega align amino acid sequence from different proteins or species. And why should we align these sequences and look at them next to each other? So it can identify conserved regions, the amino acids that remain unchanged across species and why they would be important. And then the conserved areas also indicate essential roles in protein functions, so active sites and structural domains. So we can see how different proteins function between one species and another. We can also use phylogenetic trees. So cholesterol omega also generates phylogenetic trees that show the evolutionary relationship among proteins and how protein families or functions have evolved over time. So the efficiency of bioinformatics tasks that would take weeks to manually input can now be done within a second with new software, and it's used for understanding protein evolution, identifying candidate targets for drug development, protein engineering, and functional structures. So, what is protein modeling and structural analysis? So, protein modeling is a subdiscipline of bioinformatics that predicts 3D structures of proteins and their interactions with binding partners. It helps the scientists understand how proteins function and how drugs might bind or inhibit them. So methods of structure determination, we have X-ray crystallography, which analyzes diffraction patterns from X-rays striking crystallized proteins. We also have nuclear magnet resonance, or NMR, and this uses magnetic fields to determine protein structures in solutions. So, protein modeling and structural analysis. So, we have a protein data bank, and this is a global database of 3D protein structures that we can go look at any time. And the structures can be viewed, rotated, and colored for study. So, we can get protein shapes, binding sites, and structural changes due to mutations or binding events. This helps us understand disease mechanisms, drug designs, and protein engineering. I will see you next time in protein production and industry. Thank you.